I think there should be 21 of you, 22 maybe. So once I see that many participants, I'll start the quizzes. Before we get started, does anyone have any questions like right off the bat about this material that maybe Grace or I could answer? All right.
Wait, I have a question.
Yeah.
For like circulation, since there's two parts, do you think that'll be more like prevalent on the test, like a bigger section?
Yeah, I would
Okay.
guess so. Because what that was 22 and 23.
Yeah, something like that.
Yeah, I would expect there to be a good bit on that. And honestly, I would focus mostly on your PowerPoint rather than the textbook because the textbook went very in depth on some like animal focused stuff and like still be, a little aware of that. But I think the big things he wants you to know are like the pressure gradients that like drive inspiration and exhalation and like anything having to do with like gas exchange, partial pressures, all that stuff. Like I think that's going to be a ton of your test.
Okay, thank you.
Mhm. All right, 20 people. I'm going to start it now. If you guys, the coach should be up in the corner though, if you haven't gone in yet. And don't worry about the timer. I'm going to add time to these ones. Most people got that right. So yeah, Henry's law is C equals K times P. So the concentration um, of a gas in a solution is equal to the partial pressure of it um, directly. So you got that. I think there's a way where I can view answer explanation. Okay, so I added a few of these all have definitions in here. Um, but yeah, so nothing too bad there. All right. Um, Pretty good there. Does anyone want to unmute and say what they think is false about this?
Wouldn't rate of diffusion be higher if partial pressure is higher?
Yeah, exactly. So the thing that's wrong here is the word inversely. It's directly proportional. So this is that equation that uses the Krog diffusion, whatever. Maybe I wrote it in here, but yeah, this thing right here. J equals K times P1 minus P2. K is that Krog's diffusion con or something. I honestly forget what that is, but um, since this is in the numerator here, that means that as we increase that difference in partial pressures, as we make that gradient stronger, basically, the rate of diffusion is going to be even faster, which makes sense. You guys are doing good so far. Yeah, so hemoglobin is best at binding CO2 in systemic tissues, and we can kind of think about that as being because that is where we have the biggest, you know, I guess you could say lack of oxygen, right? Is if we go to like our fingertips, they got to work all the way from our lungs down. Our blood system has to bring oxygen to those guys. So they're really in need of oxygen and they're building up CO2, right? So basically, this is kind of the Haldane effect, is what it's talking about. CO2 binds to hemoglobin really well once we're in the peripheral tissues, and that's good because that means we can bring it back to our lungs. And then hemoglobin is really good at releasing CO2 once we get to the lungs, which also should make sense, right? That's where we want to breathe out the CO2. Um, Anyone have any questions about that? Again, that's the kind of the main idea of the Haldane effect. All right, cool. Sweet. Yeah, so the answer here is type 1 alveoli and capillaries. Does somebody want to unmute and say what the difference between type 1 and type 2 alveoli are? I know someone in here is confident enough. All right, I can say it. So type 1 alveoli are going to be the simple squamous cells, like the very, very flat cells of our alveoli. And the fact that they're super flat is good because that means we have really good diffusion of the gas particles. So type 1 are what actually do the gas diffusion. And then type 2 alveoli are a specific type of cell. It's a bit more like cube-like. And those guys are secreting something called surfactant. And it is important to know what surfactant does. Surfactant is this thing um, that lowers the surface tension in our alveoli. So your alveoli have water. Water naturally likes to bind to itself. So if we didn't have surfactant, all the water would bind to itself and those alveoli would collapse. And surfactant prevents that. Good. And then, yeah, the other answer is capillaries, pulmonary veins. Although those are the things that take oxygenated blood back to the heart to be pumped out to the rest of the body, they're too big. Veins are too big for simple gas diffusion. We need capillaries. Let me add more time here. Awesome. Almost all you guys got that right. Yep. So water is dense, it's viscous, particles can't move as quickly through it. And that's why gas is better diffusing through air than in water. We have more time. Yeah, this one's a bit trickier, I would say. So, 100% you need to know all of the chemistry behind the bicarbonate buffer system. So, this goes back to some like Chem 132 knowledge of Le Chatelier's principle. So, anytime we have an equation at equilibrium, Le Chatelier's principle says that if we increase, say we increase the reactants, the reaction is going to shift to make more products to kind of like even out that balance beam. So, in the bicarbonate buffer system, we have CO2 and water going to form an intermediate, and that's carbonic acid. And that reaction takes place with the carbonic anhydrase enzyme. And then that goes on to form the, or carbonic acid breaks down into bicarbonate and a proton, so H plus or the hydronium ion. And so the hydronium ion is the big thing that is going to like lead to acidosis that lowers the pH, makes things acidic. And so the answer here is, let's see, what did we, a shift to the right, acidosis. So in hypoventilation, we can think about, we're breathing a lot slower. And if we're breathing slower, that means that we're off gassing less. So we're getting rid of less CO2 than we need to. And that means we're building up CO2. And so if we're building up CO2, and again, we can think of that as one of the reactants, then based on what I said a second ago, we build up a lot of that reactant, it's going to shift to the left side of that equation. I don't know if my arrows are reflecting right or left, but we're going to shift to the left side of the bicarbonate buffer system, meaning we're making more and more of the H plus. And that means that, again, remember pH, when it's lower, that means we have more H plus. So that's going to be acidosis. Does anyone have any questions about that? I might have one more bicarbonate buffer question in here. Okay, cool. Okay, this one is true. I didn't throw any tricks in there. So countercurrent gas exchange is when we're talking about, you can think of like the blood movement is going in the opposite direction of where the gas or the air movement is. And so, like this is really important for fish and their gills. That's like a countercurrent example. And the whole point of that is so you're kind of constantly having the blood meet different air partial pressures. And so if we do that, that means that equilibrium won't be hit. But if you can think about two things going in the same direction, over time, equilibrium will be hit, and that's not good for gas exchange. So this one is true. All right, sweet. It seems like a lot of you guys got this. This is one of those ones where it's like you either know it or you don't. I think the book goes into a little bit of depth about like how this happens, but yeah, frogs, once they're an adult, they still are going to excrete CO2 through their skin, but their lungs have then developed enough where O2 uptake, so their breathing in is going to be, yeah, through the lungs. I'll add some more time. All right, nice. Yeah, all you guys got that. So again, buccal pumping is the thing where they like open their mouth actively so that they can kind of draw in air through negative pressure and then breathe it out. And that lets these fish breathe while staying still. And then ram ventilation is a passive process. So that's going to be, they're swimming, and that's enough where you have this oxygen dissolving through like past them. So that one's a passive process. Nice. You guys are doing well. So yeah, vital capacity. I don't know if I threw in the photo, but yeah, this photo right here. Um, Make sure you know this very well. Resting tidal volume is the amount of air in a normal breath when you're not exercising or anything. Inspiratory and expiratory reserve volumes are kind of like what you could tap into if you need to get more. So if you start like working more breath in or exhaling a lot out, vital capacity was the answer to this one. That's going to be if you add up your basic tidal volume and the maximum amount of inspiratory reserve and expiratory reserve. So it's like the most difference you could get pretty much. And then residual volume is whatever is beneath that line that like you can never really prove out. Cool. All right. So Um, this one's very important to know. There's different ways that our body can kind of modulate our autonomic nervous system so that we can either breathe more or breathe less, depending on what's going on in our blood. And so the primary thing that is going to tell us to like maybe, hey, we need to breathe a lot faster to get rid of this carbon dioxide. That's going to be central nervous system chemoreceptors in the medulla oblongata. Um, then a little bit beneath that, but still a little, it still happens. There are peripheral chemoreceptors, and I think I put the two things down in the carotid bodies and aortic bodies. You may or may not need to know that. Those also have an impact, but not as strong. And then, like the last thing that has a very small impact, and this is only if we're in like a really bad state, is the actual like partial pressure of oxygen in the blood. But yeah, the big thing to know is that the medulla oblongata is what's going to signal to our heart to start like beating faster or to our lungs to make us breathe more. All right. Does somebody want to say what's false about this? If you got this one right.
Uh, I think birds store air in a second compartment before it goes across the lungs.
Yeah, that's another. There's a few things that are unique about them. That's one of them. The thing that I was kind of getting at with this question is that They do have a unidirectional flow system, so that's true, but it's not called a countercurrent. Instead, it's called a cross current for birds because birds have this unique thing. Maybe I have a photo. Okay, good. Cross current is like lets them be super efficient with gas diffusion because we have blood flow that's perpendicular to the air movement, and that's unique. That's unlike I think most other animals. But yeah, good thing to know. Cross current, not counter current for those guys. I'll give more time for this one. All right, I'll talk a little bit about this one. So the answer here is C, parasympathetic going, uh, parasympathetic nervous system is going to innervate our lungs by releasing acetylcholine, and that's going to cause our lungs to constrict. That's a little bit of a weird one. We don't think about that too often because really we're not going to be constricting our lungs too much. But when we're resting, we don't need to have them as wide. And then the other answer that is not here, but the sympathetic nervous system innervates our bronchial smooth muscle by releasing norepi, and that's going to cause bronchodilation, which, if we're thinking of like an EpiPen gives straight epinephrine to a person because they can't breathe, right? And that's going to, loosen up that smooth muscle, allowing for more gas exchange because that's the ultimate thing. Like if you're running away from a bear, also sympathetic nervous system, we want to be able to breathe a lot more. So, does anyone have any questions about that one?
Wait, yeah, for the heart, isn't it like the opposite? Or am I confused?
Yeah. So you're talking about like uh, whether we're
Like,
speeding up or slowing down the heart rate?
yeah, yeah, like doesn't acetylcholine slow the heart down
Oof.
or like relax it? I'm not sure.
Um, Grace, do you know this one?
I'm not quite sure.
Okay. Um,
I'll look it up.
yeah, I'm not quite sure. I'm sorry. I think it's like I forget exactly how that works, but there's like the different receptors. I know you have like the different beta receptors of the heart that can either lead to faster heart rate or slower heart rate. But I can't remember if it's acetylcholine or a different
Okay,
transmitter. Sorry.
all good.
Seems like you guys are pretty good with this one. Yeah, so this is that one slide in your book. I don't think I Yeah, I didn't really have the photo, but the photo where you see the like lungs atmosphere or the lungs pressure in millimeters mercury, then the pleural spaces pressure, and then the atmospheric pressure. Definitely understand what's happening there and how there's like even a small difference in pressure is what drives the entire gradient. So like understand that the pleural space is going to be a little bit lower. Um, I think it's like 4 millimeters mercury less than what your lungs are, and that's what's holding that like negative pressure suction that's really like keeping the lungs open. And then yeah, also understand inspiration and exhalation. And so in this instance, when the lungs drop 1 millimeter mercury, that's what draws in air and lets us, inhale. So yeah, here's another one with the bicarbonate buffer. Oh, I think I wrote this one wrong. I'm sorry, guys. I meant for it to say leftward. That's on me. So don't worry about what they say is right or wrong. I'll talk about it after. Yeah, ignore that. My bad. So it was supposed to say leftward shift, and if we're shifting left, the only thing of these answers that would do that would be a sudden increase in the blood H plus concentration. Again, that goes back to Le Chatelier's principle, where if we increase one thing, so if we increase the product, it would shift to the left to make more of the reactants. All these other things, a decrease in respiratory rate, exercising, or these two would lead to a rightward shift, right? A decrease in respiratory rate would build up CO2, meaning we shift to the right side. Exercise, increased metabolic rate. Again, that's going to be producing CO2, and so we're going to shift to the right. And then this one down here is a trick answer. So increasing the amount of carbonic anhydrase in the blood would actually do nothing. So if you add a catalyst to a reaction at equilibrium, it's going to equally speed up both the forward and the reverse direction of the reaction. So that one's going to do nothing overall. But yeah, is that clear for everyone? Does anyone have any questions about this stuff? Cool. All right.
Okay, does anyone want to share why this one was false? Anyone?
Isn't it like temperature dependent?
Yes, so when the sex determination occurs for animals, some animals such as turtles and usually reptilia too, other reptiles, it's done through temperature sex determination, which is where the like different temperatures such as hot or cold influence what sex the animal will be. Okay, any questions on that? Okay, so the answer to that one was A, D, and E. So basically, pre-mating processes are things that allow the mates to meet each other. So it's things such as acquiring resources, monitoring environmental cues to know when the right time it is to find a mate and like when to start making things. And then the things that you do for post-mating processes are what you do to ensure that the baby will be viable until and will create a successful offspring, if that makes sense. So that would be development of zygotes, evasion of predators, and possible diversion of familial time. So making sure that the offspring survives. Any questions?
Wait, can you explain familial time a little more?
Okay, so
I'll
possible
pause this.
diversion of familial time is like the different roles that parents of an animal may take or share to make sure that the fetus is viable. So like for example, if it were like a male cardinal, he typically would go out and hunt for food, and bring it back to the nest. While the mother, on the other hand, would be in the nest, like making sure that the baby is fed, stuff like that, like the actual feeding and everything, like for the baby cardinal. Does that make sense?
Thank you.
It's kind of like a hunter-gatherer thing.
Okay.
That's kind of what I thought of it as. Any other questions? Okay. Awesome. So the hormone that causes the most or contributes the most to ovulation is LH. It's always indicated by that large LH surge right before ovulation. Any questions there? Okay. Just like
Damn, they gave 3 minutes for this one. We can cut that early.
okay, so ignore the grading, but the correct order for specifically the menstrual cycle as a whole, combining the uterine and the ovarian cycles, is the menstrual phase, the follicular phase, and the luteal phase. And then ovulation is also in there. Okay, so LH will stimulate the thecal cells, and FSH will stimulate the granulosa cells. So I'm trying to think of a good way to remember this besides just straight up memorizing it. Um, I think
FSH and granulosa both have S's in them.
yes, that's a good one. LH and theca both have H in them. That could be useful. I don't know. If that
Yeah.
helps you, fantastic. Yes, this one is true. So LH Leydig cells, they both have an L in them. FSH Sertoli cells both have an S in them. But the main thing that makes me remember the difference is Leydig and LH always go together because they both have L's. And then Leydig cells go on to secrete testosterone, and FSH and Sertoli cells just support the sperm. So yippee, good job. Okay, so in order, the correct answer was B, which is or A, sorry, which was in order B, C, D, A, and E. And I pulled this from your lecture, your chapter 17 lecture PowerPoint. There's the HBG axis in there, and so basically in order, it's first myometrial contractions force the fetus against the cervix. Then oxytocin secretion becomes more active, and then blood oxytocin levels increase, and oxytocin stimulates smooth muscle contraction, and increased oxytocin triggers prostaglandin release. If you guys have any more questions about that, I can definitely talk more after the rest of the questions because I know this one is definitely more difficult to understand. But I think the biggest thing you need to understand is it's a positive feedback loop instead of a negative. Do you have any questions right now? Okay. Okay, so the correct answer for this one was false. Does anyone want to tell me what stimulates lactation? What hormone?
Isn't it oxytocin? I think.
Yes, it is oxytocin. So oxytocin is what I think of as the main thing for give like the main hormone, one of the biggest hormones responsible for giving birth and lactation. So I think about that as associated specifically with pregnancy and post-pregnancy. And that's kind of what I think of it as, just to make sure I remember that. Okay, good job. Okay, so the correct answer for this one was B and D. So cardiac muscle is a little unique. It has the visible striations, kind of like our skeletal muscle, but it's not innervated by the somatic nervous system like our skeletal muscle. It's autonomic. So basically, it's just automatically always beating. If it were somatic, we'd have to tell it when to beat, and that could be really complicated and not sufficient. So, and then also it is a calcium regulated cross bridge. If we were thinking of unstriated, that would be your smooth muscle, and it would be innervated by the autonomic system too. Are there any questions on that one?
Please get this one right, guys.
This was just a gentle reminder.
Yay.
Yay. So it's the answer is C. From the heart, oxygenated blood is that's what I remember. So from the heart, arteries carries oxygenated blood. And then to the heart is veins carrying deoxygenated blood. Slay, good job.
Does anyone know the arteries or veins that do the opposite, that like the vein that carries oxygenated blood or the artery that carries deoxygenated blood? Because you'll probably talk about that on the test too.
Isn't it pulmonary?
Hmm. Yeah, the pulmonary. So it's care you want to be careful to say that like the pulmonary vein is actually an instance where we have oxygenated blood in a vein because it's going um, back to the heart, but it's coming from the lungs. And pulmonary artery is the opposite.
Okay, so again, I don't know if it's gonna grade that correctly, but the answer is it in order. It goes P, Q, R, S, T. So like the alphabet. And from P to Q, based off of what Dr. Parker put in your lecture slides, P to Q is atrial systole. R to S is isovolumetric contraction. S to T is ventricular contraction. T is isovolumetric relaxation, and T to P is ventricular filling. Does anyone have any questions on that? I think the easiest way to get this one for the exam, because he will ask you something about it, is to definitely like drill it into your head, like have that sequence memorized to a T. Okay, so the correct answer was D. So the initial depolarization comes from the sodium funny channels, which are like a little like they're called funny because they behave a little differently. And then the continued depolarization comes from the sodium channels depolarizing to open a certain type of calcium channel. And this is the transient type calcium channel. And that allows us to depolarize the threshold for the pacemaker cell. And once we get to threshold, that L-type calcium channels come in and kind of maintain that depolarization. Any questions on that?
Wait, so when the L-type channels open, is it implied that the T-type channels close?
No, I think they're still open, but the exact timing is probably in your notes, but they kind of slowly close.
Okay.
Okay. Slowly. Okay, so the correct answer was B. This one was particularly a definition pulled from your notes, but a neurogenic heart has cardiac muscle contraction initiated by neuronal signals. So that would be like an example of our heart. Any questions on that one, or are we good? Okay, so again, it's not grading it correctly, but for an action potential in a neuron, it has a rapid depolarization and repolarization phase, and it lasts about 1 millisecond. However, cardiac action potentials can be funky and have a plateau phase, and that's due to prolonged depolarization, usually from the L-type calcium channels, and that makes it last around 100 milliseconds. So I think the biggest thing that I remembered was the cardiac action potentials have that long plateau phase.
Um, so if I were to answer that the cardiac have a long and dull shape, while the neuronal has like short and sharp shape, would he have accepted that for full points, or would he want me to go more into detail?
Definitely more into detail, probably partial, but like maybe say like what makes it like have that long and dull shape, like go into why that's there. Maybe more specifics on the shape too.
All right, thank you.
No problem.
I just want to clarify. So like when it plateaus, that's it repolarizing, right?
So the plateau phase is a sustained depolarization, and that comes from the L-type calcium channels.
Oh, okay.
Okay, so the correct answer was length and radius. That's what influences the flow rate. It's not mass and charge, it's not width and height, and it's definitely not radius and diameter since those measure kind of the same thing. But usually radius is proportional to resistance. So or inversely proportional to resistance. So the larger radius you have, the more it's going to evenly or the better it's going to flow because there's less resistance. And for length, that also plays a factor too. Like the length that the blood is traveling makes a difference. So I would review that mathematical equation just to look at the factors that go into it. Are there any questions? Yes, good job. Baroreceptors do sense pressure. And one thing that, if we had more time, I would go into a little bit more depth on is make sure you understand the baroreceptor reflex, why it's done, and like what the steps of it are. And that's at the end of the chapter 25, part 2 notes. But yeah, good job.
I think that was the last one.
Yes, that was the last one.
Let's see. 65, that's pretty good. But you can shoot for a little bit higher tomorrow on the test, but that's pretty good. Um, I'll stick around here for a little bit if anyone has any questions. Um, but yeah, other than that, you guys are good to go. I should have this recording posted later tonight as well. So if you want to come back and review some of these questions, feel free to. But at this point, I would say your best thing to do is like hammer in those PowerPoint slides. really focus on that stuff. And especially like the big picture stuff, the bicarbonate buffer, the pressure gradients, all of really all of repro is important. And then EKGs, vascular system, that stuff. That's what you should put your time into between now and your test. Yeah, thanks for coming, guys. I'll feel free to ask any questions if you have them.